looks like. We'll be talking about more about what's going on inside and how exactly they contract. This isn't physiology, so I'm not going to test you too hard on like the mechanism of contraction. I'm sure some of you are pretty interested in it, so I'll explain it, but I'm not going to test you on the specifics. I will test you on the specifics of what's the things inside of a muscle fiber. So we're going to learn about weird names like A bands and I bands and H lines, and why those things matter. Why do we bother naming all these weird things inside of a muscle fiber? And in lab, we'll see what that looks like. We'll see what that looks like, and we'll start looking at actual whole muscles in the body, just like we look at whole bones before we actually look at bone tissue. Cool? All right, we left off on slide. Yeah, here. Slide 16, 15, 17. What is the plasma membrane of a muscle fiber called? Sarcolemma. So, draw it as red. All right, here's our sarcolemma. What is the connective tissue that wraps around every single muscle fiber? Endomysium. So that is outside of the sarcolemma, wrapping around it. We have our T-tubules. Diving deeper in at regular intervals. To help spread electricity deeper into the cell. Oh, I forgot to draw nuclei. They're multinucleate, and the nuclei are pressing up against the sarcolemma. There's no pattern to them, there's just a lot of them. They're just all over the place. The nuclei. Oh yeah, and then just the inside of the cell, everything inside of the cell is called the sarcoplasm. All right. Oh, I'm missing sarcoplasm. What should I use? Between each T-tubule, there's many sarcoplasmic reticula. And these sarcoplasmic reticula, where are they found? They're found wrapping around. They're found wrapping around those myofibrils. You can see there's one here. There's another. There's another. They're wrapping around every section of a myofibril. Okay. Any questions on all that? Those are things we've already gone over. All right. What is a myofibril made of? Now we're going to start talking about the specific proteins that we need inside of our muscle fibers, the proteins that help us contract. There's several. Proteins are really important for muscle fibers. Proteins are important for every cell, but we know that proteins are really important for muscles. So, what are they made of? They're made of two different filaments. There are thick filaments, which are thicker, and there are thin filaments, which are thinner. Thick filaments are made of a protein called myosin. Whenever you see a word end in 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 biology, it's typically a protein. Just like, well, collagen's in EN, but collagen, myosin, actin, those are all proteins. So myosin, shown there in that purple color, that is our thick filament. Thin filaments are primarily, there's actually other things associated with it. Thin filaments are primarily made of actin. So how does that look like? How does that look in our drawing here? Within a set of T tubules, so here's our myofibril. Within a set of T tubules, one myofibril will have a thick filament, or a set of thick filaments. That's our thick filaments. And then the thin filaments are they're spanning that in between. They're kind of interlaced. Kind of interlaced in between. You have thick filaments, we're going to say at the center of this region, and thin filaments are on the sides of this region. This region, which repeats over and over and over again all along the myofibril, that region is called a sarcomere. That region is called a sarcomere. So the whole long thing is a myofibril, but this repeating unit here, here, and there, this repeating unit is called a sarcomere. It is the sarcomere that contracts. Sarcomeres get shorter during contraction. It's because of this organization that we see striations. So we can't see individual protein molecules with our light microscopes, but because they're all organized so well, we can see the banding pattern. Just like when DNA condensed into chromosomes, we can see the chromosomes, but we couldn't see an individual DNA molecule, but we could see them all clustered together. Same idea here. All the proteins are organized, very organized, so we see the banding pattern. Because of the organization of these thick and thin filaments, because of the organization of sarcomeres.
So, based on the drawing, the myofibril doesn't go through the sarcoplasmic?
Oh, based on the drawing, does the myofibril go through the sarcoplasmic reticulum? Imagine like a really long. This is the myofibril. Here's the sarcoplasmic reticulum wrapping around it. It's around it.
Can you say that again?
Sarcoplasmic reticulum wraps around the myofibril.
So it's like gravity and pulling itself, and that's how it gets back.
The sarcoplasmic reticulum itself doesn't move much. I mean, it will move with everything else, but it's not the movement of the sarcoplasmic reticulum that matters. It's the movement of the sarcomere that matters when the muscles contract. The sarcoplasmic reticulum is just surrounding those filaments, those thick and thin filaments.
So it doesn't necessarily move.
Whether the sarcoplasmic reticulum moves or not is irrelevant. We'll see that it helps cause movement of the filaments, but it's there wrapping around the filaments. It's wrapping around the filaments.
And the sarcomere is like a section of the myofibril?
Yeah, sarcomere is a section of the myofibril. If this is a muscle fiber, this is a myofibril, and we had this divided up into even sections. Each section is a sarcomere. Sarcomere, 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 sarcomere. And this organization repeats over and over and over again. So get used to how this looks. This repeats over and over again.
Wait, so where would the sarcomeres be? Like on the drawings?
Where would the sarcomere be? Sarcomere be? This is one sarcomere. And this repeats over and over and over again in between T tubules. From Z disk to Z disk, and we're going to learn what the Z disk is coming up.
So the T-tubule just lays over the sarcomere?
The T-tubules are, I mean, this is in 3 dimensions, so the T-tubules are kind of like around in this plane. This myofibril extends all the way through the muscle fiber. So I know you can't see it beyond this T-tubule, but it goes beyond it. It goes beyond it. So this repeats over and over and over again, these sarcomeres. And it's hidden here, but they would kind of join together in a way. Okay. Right. Okay. So within, what is each thick filament made of? I mentioned myosin, and a single myosin molecule looks like this. It's got a long tail. It's got this bendy hinge region, and it's got these 2 heads. We're going to focus on these heads because they're functionally very important. So if I were to try to draw like a single molecule of myosin here, it would look like here's its tail, here's the hinge, and then here's the head. Tail, hinge, head. And you have a lot of these, all of this bundled together. Yes?
Could you possibly make like a bigger one? Because I can't.
Yeah, sure. All right. Tail, hinge, head region. And you have a lot of these bundled together. So here's a tail, hinge, head, tail, hinge, head. All these bundled together make up a thick filament. So this is one myosin molecule.
Each head has its own tail. It doesn't feed off of each other.
Do they feed off each other?
Like, it's not like
They're all staggered. So they're all
individual.
Yeah, so this head has its own tail, this one has its own tail. And it's all 3-dimensionally wrapped around this whole, and they form the whole filament. I keep mentioning this head region. This head region is important because it's very reactive. This head region wants to bind with the thin filament. It's really attracted to it. There's this other protein called titin. Here's that middle of the thick filament. Here's the thick filament extending out, and then here in green, that protein here is called titin. Think of this as like a bungee cord. It's just there to hold things in place. You don't want your rails to go off. You don't want your train to go off the rails. This is to make sure that it only moves in this motion and not in a different motion. So this protein titin is there to just to stabilize myosin. And that's really all I want you to know about it. Titin is, what color is? Titin would be right here. Help stabilize myosin. Okay, so those myosin heads are really attracted to the thin filaments. Specifically, they're really attracted to actin. So an individual, well, shown here, and I'll redraw it. All these circles, all these red circles, Those are actin molecules. And they're arranged like on this skewer-like thing into a filament. And these myosin heads are attracted to them. What allows actin to be arranged like this is that there's this, you know what, don't worry about that molecule. Yeah, you know, I wrote that. Okay, so there's this molecule, it acts like a skewer. So imagine there's this rod that goes all the way through. What holds the actin molecules in place in a filament-like arrangement, it's called nebulin. So actin has nebulin to hold it in place. Myosin has titin to hold it in place. So titin and nebulin are just support molecules. They're just all help things stay in place. These are all proteins, by the way. Two more proteins that you should know. Troponin and tropomyosin. Troponin, how do I describe this? Also found on the thin filament. So to make up the thin filament, we have nebulin holding it all together, actin bound to nebulin. Surrounding actin is this longish molecule called tropomyosin. Tropomyosin. The analogy that I got from Crash Course, imagine myosin and actin, they're like two kids at a school dance. They want to get really close together. So they're really attracted to one another. Tropomyosin, along with troponin, they're the chaperones. So you just, it's like put that grapefruit in between them, you know, or whatever other fruit. So they can get close, but not too close. They can get close, but not too close. So myosin is really close to actin, but tropomyosin is in the way. Yes?
So where's titin?
Titin's over on the edge. It's just, so here's the thick filament, titin's over here. Titin's a supportive molecule, titin's here in green. So right now I'm just looking closer in here. So here is tropomyosin, and troponin is, it's found associated with tropomyosin all along it. Okay, so you're at the stance, they want to get close, but they can't get too close because tropomyosin's in the way. I guess I'll just say this. What happens is, when contraction happens, a signal binds to troponin, and when that signal binds to troponin, it moves tropomyosin out of the way, so that myosin can grab onto actin more strongly. So this is why I call troponin and tropomyosin regulatory proteins. They regulate the interaction between actin and myosin. If you don't have a signal, myosin can't bind to actin strongly. If there is a signal, and we'll talk about what that signal is, if there is a signal, then then troponin is distracted, pulls tropomyosin out of the way, and the two can bind together. We'll talk more about that when we get to everything else. Okay. So when everything comes together, because of the arrangement of the sarcomere, we get striations. How can I draw this? Okay. The very center line that's holding all these thick filaments together, Oh, sorry, I'm starting with the wrong place. At the ends, let's go back to the thin filaments. Where the thin filaments are, what's holding the thin filaments together, it's actually a zigzaggy band. I drew it as straight, it should be zigzaggy. There's a zigzaggy band on either side of the sarcomere. So there's a zigzaggy line here, there's a zigzaggy line here. Fortunately, it's called the Z line. So look for the zigzaggy line, that's the Z line. The Z line is the boundary for each sarcomere. It's at the ends of where the thin filaments join. So here we have a Z line, here we have a Z line. So this is the border of one sarcomere. So these are thin filaments for one sarcomere. These are thin filaments for the next sarcomere over. So this thin filament is continuing from over here. This thin filament continues over here. The Z line's in between them. Titin is connecting to those Z lines. So here's the myosin, the thick filament. Here's titin. Titin goes all the way to that Z line. What do you think happens to the Z line during contraction? Does it move out? Does it move in? Does it not change? The Z line will move in. Sarcomeres get shorter during contraction, so the Z lines move towards the middle. What's the middle? That's the M line. The M line is the very center of our sarcomere. So here's the M line. So Z lines move in towards the M line during contraction. If you have an electron microscope, this is an electron micrograph. If you have an electron microscope, you can actually see the Z lines. These really thin lines here. You won't be able to see them on your light microscopes. It's too small. But you can on an electron microscope. Right. And then we have these alternating light and dark bands. The lighter bands, those are called I bands. So what on here is an I band? It's lighter where there's only actin, where there's no overlap with the thick filaments. So where is there only actin? Here's the end of the thick filament. So from here to the next start. So it's kind of crossing from one sarcomere to the next. This is an I band. From the end of one thick filament to the start of the next thick filament, crossing over sarcomeres. That's what an I band is, where there's only thin filament, there is no thick filament. So here's an I band. End of one thick filament, start of the next thick filament. This is the I band. So the Z line is in the center of the I band. The Z line is in the center of the I band. What do you think sorry.
Where is titin in that?
Where's titin in this? Titin would be connected from the thick filament to the thin filament.
So that's included in the I band too, or no?
Yeah, titin would be found in the I band. Yes, titin would be found in the I band. What do you think happens to the I band during contraction? Lengthens or shortens? If the Z line's moving in, you're reducing this space and increasing the overlap. So you're minimizing how much unoverlapped actin there is. So yeah, it gets smaller. Wherever you find the thick filament, because it's thick, it's darker, Wherever you find the thick filament, that's where you find the A band. So this whole span here to here, that's known as the A band. I and A band, they stand for something. Isotropic, meaning uniform, and anisotropic, meaning polarized, like sunglasses, some sunglasses. The way that I remember it, I band is the lighter band, A band is the darker band. I can't remember isotropic and anisotropic, but maybe it's an easier way to remember it.
What is the Z line made out of?
What's the Z line made out of? It's actually made out of a special kind of actin. It's called actinin. Within the A band, again, we can't see this with our light microscopes. Within the A band, that's where the M line is. And if you look really close, it's slightly lighter in the middle. You can see here's the zone of overlap, where you have an overlap between thin and thick filaments. So this region is the zone of overlap. And you have it on either side. This overlaps twice. But there's a region where there's no overlap, where it's only thick filament. The region where it's only thick filament, that's known as an H zone. Lots of letters here. So if you look really closely, you'll see that there's a very light line in the middle of the A band. Slightly lighter. It's still thick filament, so it's going to appear dark to us, but it's lighter than what's around it. It's because there's no overlap. That's known as the H zone. H is German for bright. However, what do you think happens to the A band? Or sorry, let me back up. What do you think happens to the H zone during contraction? Bigger or smaller, stays the same? Here's the thin filament. It's going to move in, right? It's going to get smaller. H zone gets smaller. How about the A band? What does A band represent? What's found in the A band? Thick filament. Only the A band is found in the thick filament. If our frame of reference for a sarcomere, if this is what we're calling the center of the sarcomere, and things are moving inwards towards it, thick filament doesn't move. It stays the same. So to recap all that, what's happening here? All these myosin heads, when they get the chance, when they get the signal, when they can get closer to the actin, what they'll do is they'll grab and pull on the thin filament. So on this half, these heads are grabbing and pulling towards the M line. On this half, they're grabbing and pulling towards the M line. So everything moves in towards the M line.
They pull it towards itself.
Pulling towards itself.
Okay.
Pulling towards itself. So you can see how the sarcomere gets shorter. Here the Z lines move in. Z lines started here, Z lines move in. So that means the I band gets smaller, the H zone gets smaller. The zone of overlap increases. I forgot to mention that one. Here we have some overlap, here we have more overlap. Zone of overlap increases. What does not change is the A band. I band gets smaller, A band stays the same. Yes.
Did you say that the I band spans the length of the sarcomere?
The I band the I band does not span the entire length of the sarcomere. The I band crosses from one sarcomere, it starts here and goes to the next
So it
sarcomere.
connects one sarcomere to the next?
Yeah, the I band connects one sarcomere to the next. That's a better way of saying it. because we're saying that our borders of the sarcomere is this, but the I-band is this. So it's crossing from over one sarcomere to the next.
So at the end of one and the beginning of the next?
It starts at the end of one and ends at the beginning of the next, yes. Yes. Okay. Oh, this is known as the sliding filament theory. Theory in science, if you're not familiar with that word in science, theory is not a crazy idea. Theory means something that's well established. So this is our best explanation of how muscles contract. The reason why we still call it a theory is we don't have any direct evidence that this is how it actually happens. We have lots of evidence pointing towards it. We don't have anything concrete. That's why it's called sliding filament theory.
It's called a what?
Sliding filament theory. They slide past each other. For muscles to contract, literally muscles get shorter, we need proteins to slide past one another. It's not that you're losing protein, which is what a theory could, or another hypothesis could be, that you're losing protein, but that's not what we see. What is likely happening is this. Let's turn the brightness up, or flash off brightness up. Okay. So, you can see that muscles are really organized. Skeletal muscles are really organized. And we're going to see that cardiac is organized in the same way. Cardiac muscle has the same type of organization. That's why they have striations. We've gone over how whole muscle is covered by epimysium. Fascicles are covered by paramysium. Fibers are covered by endomysium. Within a muscle fiber, you've got myofibrils. So all these little things are myofibrils. Myofibrils are bordered by T-tubules, surrounded by sarcoplasmic reticula, and organized in sarcomeres. And sarcomeres have myosin and actin and all these other proteins and help them work properly. All these multiple levels of organization help them work efficiently, provide a lot of tension to do things like move your body. This?
The whole picture set.
This whole picture is cardiac muscle has a similar arrangement.
But not this exact.
It wouldn't just because cardiac muscle is branched. This is multinucleate. Cardiac muscle has only one nucleus. If we just look at the arrangement of a single myofibril, that is very, very similar. Very similar. There's like minor changes, and that we're not going to get into. We're in physiology. Then smooth muscle is a completely different arrangement. Raise your hand a few more times. Hands on 3. 2, 3. Real quick, talk to someone behind you or in front of you. Talk to someone behind you or in front of you. One more time, please. your hands. 1, 2, 3. Three said letter. 1, 2, 3. E. Yeah. We're getting more overlap with the thin filaments and thick filaments. So the thin filaments are moving out of the I-band. A would be incorrect because this is saying that the M line is moving. The M line doesn't move in our frame of reference. We're calling this the center of the sarcomere, the M line. And based on this frame of reference, everything's moving closer to it. The thin filaments are moving closer to it. So yeah, B is the best answer. It's definitely not moving away. We're contracting. Like soreness after working out. Like you haven't lifted in a while and you feel sore like the day after, or the day or two after. Myelin sheath is for like protection. This isn't for protection. It is surrounding it like kind of like that, but it's the one delivering the signal to tell trophin to move out. It has the calcium is the signal. Anything more time? 10 seconds. All right, hands on 3. 1, 2, 3. On 3, say the letter. 1, 2, 3. A. Yeah, it's A. If we're damaging the Z line, thin filaments directly connected to the Z line, so we're getting damage to that. So, thin filament. Okay? Okay. One more thing to add to our diagram here. How do we actually tell our muscles to contract? Now we can put everything together. We need a neuronal signal, a signal from neurons, nervous system cells, to tell them muscle fibers to contract. So, all right, we know this is sarcolemma endomysium. I have to raise this part. It's always in 3s. Thank you. All right. So going through the endomysium. So remember, this is in 3 dimensions, so I can't draw it coming around. But you have your endomysium here, but going through the endomysium, you have the end of a neuron. So here's the end of a neuron. The end of a neuron is called an axon terminal. It's called an axon terminal because the extension that brought it here is called an axon. The extension, I can't see it very well. Here's the axon terminal, and then here's the axon. Axon, axon terminal. Axon, axon terminal. The axon terminal contains chemicals, neurotransmitters, contains chemicals that are actually stored in vesicles. How do you think it releases it? Exocytosis. So we release these chemicals, neurotransmitters, into this gap. What these chemicals then do is that they'll bind the special receptors, special membrane proteins, receptors. When these neurotransmitters bind to the receptors, that leads to a change in the cell, in the muscle fiber. All right, some terminology here. Axon terminal is where the neurotransmitter is released from. It's part of the neuron. The part of the muscle that has the receptors, because it's not the whole sarcolemma, it's just that part of it. And there's going to be multiple parts. But this region here, where we have the receptors, that's known as a motor end plate. Motor end plate. Together, or let me mention one more thing. This space here, the space where the chemical travels across to get to the muscle fiber, what fluid compartment is it? It's interstitial fluid. It is interstitial fluid, but it has a special name. This specific region is known as a synaptic cleft. Synaptic cleft. That space is known as a synaptic cleft. Altogether, the axon terminal, synaptic cleft, and motor end plate, that's what, that's all referred to as a neuromuscular junction. The junction between a neuron and a muscle. Neuromuscular junction. It's way better on my screen. So, a lot of you have already looked at this under the microscope. You saw something like this. I would have to be specific if I'm referring to the axon terminal, this cleft, or the motor end plate, or the neuromuscular junction as a whole. The whole region is called the neuromuscular junction. Just the part of the neuron is the axon terminal. Just the part of the muscle fiber is the motor end plate. Yeah, axon terminal, that term only applies to the neuron. Motor end plate, that term only applies to the muscle fiber. If you're referring to the whole thing, the whole region, that's a neuromuscular junction. So it's both the region of the neuron and the region of the muscle.
So the endomysium and the sarcolemma, is it like continuous or
The endomysium and the sarcolemma. So the endomysium is still here. So here's the endomysium. We're in 3 dimensions, so imagine it's going around the terminal.
Okay.
And then this is now closer to the sarcolemma.
Okay. And so how the interstitial fluid is from the sarcolemma?
There's interstitial fluid between the sarcolemma, motor end plate of the muscle fiber, and the axon terminal of the neuron. And that interstitial fluid is known as the synaptic cleft. The cleft is the space that has fluid. Yes, it's all the same. It's fluid, space, same thing. What type of fluid is in the cleft? It's a mix of water and ions and whatever else is there. That fits in the category of extracellular fluid.
Will that be on top of that?
It's the type of, just go with interstitial. It's the type of interstitial fluid in that cleft. All right, we'll talk more about this lab. You've all got your, what do you call it, muscle package ready? All right, I'll see you all now. And notes. And then we're going to do our activity. Well, not activity. It's lab check 8. What, 7? 7. I know we haven't formally gone over the muscles, but this is going to be our introduction. You're going to be learning about all the muscles from everyone else. Okay? So that's what we're doing for the rest of today. Probably going to take a good amount of time, I'm sure. Any questions before we get started with this part of class? We left off here. Great, thanks. Is this too dark? All right, let me redraw what we're looking at here. So a neuron, its cell body, it's somewhere over here. And then it has long extensions called axons, and we'll talk about what those are later. It has long extensions called axons, and these axons can branch. So here's a bunch of branches. The ends of these branches, those are axon terminals. And each axon terminal terminates at different things. It communicates with different things. So you can see different axon terminals here. Here's one, here's another. There's another one off the screen. Those are different axon terminals from that neuron. A muscle fiber will be found at the end of one of those. So let's say here's one muscle fiber. This region of the muscle fiber where the axon terminal meets, that's the motor end plate. The gap in here, that's the synaptic cleft. It's literally a space with fluid. And all this together, this whole thing together, is referred to as the neuromuscular junction. Let's write it out. Neuromuscular junction, where a neuron communicates with muscles and gives it the signal to contract. To recap what's happening here, at the axon terminal, you've got vesicles full of neurotransmitter. I'm not going to test you on what it is here to see it going, but you have neurotransmitter in vesicles. The vesicles get released, or the exocytosis happens, releasing the neurotransmitter into the space. So we have neurotransmitter in the space. Ignore these yellow things. We won't cover that. These red things get released into space, and they bind to receptors, membrane proteins, called specifically receptors. And it's binding to the receptors on the motor end plate that initiate the signal for contraction. Specifically, what happens when these receptors get activated? If we zoom in to the motor end plate, so let's say here's the motor end plate, the sarcolemma. Let's say here's a receptor. A receptor gets activated with a neurotransmitter. Neurotransmitter binds into the receptor. What then happens is that this receptor will change shape through a process we're not going to cover. And when that receptor changes shape, it activates a protein channel. This is a special membrane protein. It is both a receptor and a channel, a transport protein. So when the receptor is activated, the channel opens up. There's a gate to it. The channel opens up. The gate opens. Why does that matter? If you open the gate, things can go in or out of the cell. We're regulating what goes in and out of the cell. The thing that we care about is sodium ion. You might not know what sodium ion, I mean, probably heard of it before, but just looking at how I've written it out, Na+, what can you tell me about the ion? Positive charge. This positive charge enters the cell. It rushes into the cell. Movement of charge, that's what current is. That's what electricity is. Movement of charge. So if we have sodium ion going into the cell, now the muscle gets an electrical current. Electricity is flowing through it literally. Everyone with me so far? I'm not going to test you on this part. This is just for your own edification. Care to know. I'm not going to test you on this part. If you want to know what happens from here, acetylcholine, the neurotransmitter, binds the receptor. I guess this is something you should know. I'm not going to test you on the specific things, but for example, when that electricity comes in, we want it to spread in more evenly. Here are those T tubules. The electricity can spread more evenly into the fiber because of the T tubules. That's why we need T tubules. So you should know that. The T tubules help spread the electricity deeper in. And remember how the T tubules are at regular intervals? They're at the borders of all the sarcomeres. And remember how the sarcoplasmic reticulum also spans that area? When the electricity spreads more evenly deeper in, it also gets to the sarcoplasmic reticulum. What does the sarcoplasmic reticulum do? When electricity gets to the sarcoplasmic reticulum, that tells the sarcoplasmic reticulum to release calcium ion. Calcium ion is released into the sarcoplasm. And calcium ion will then bind to troponin. Troponin, when bound with calcium, moves tropomyosin out of the way. When tropomyosin is out of the way, what happens next? What molecule interacts with what molecule? Myosin binds to actin, and that causes a power stroke. It's this whole chain of events, starting with neurotransmitter binding to that receptor.
It's happening
Really fast. It happens really fast. Milliseconds. So that's why we need T tubules to help spread the electricity. We need sarcoplasmic reticulum to help release calcium ion. Calcium ion is the true signal that directly causes contraction. So neuronal signal indirectly causes contraction. Calcium ion directly causes contraction. What else do I want to say about this? Oh, like whenever you see movies and shows those dart tips that paralyze people? I forget the name of it. What's it called? No, not tranquilizer, that's something else. Like the one that literally paralyzes. It's a South American, it's from South America, it's a poison. I can't remember the name. Okay, anyways, whatever that poison's called, what it does is it blocks this receptor. If you block the receptor, now the neurotransmitter can't bind, you can't control your muscles. Neurotoxin is just a generic word for a toxin that hurts your nervous system. It's a specific word for it.
If there is like
Kill me.
problem,
Sorry?
if they block it, there is no way to activate it.
If you what?
If it's blocked and
If you block this receptor, you can't activate the muscle.
no, you cannot activate again.
Can you activate it again? It depends on how permanent that block is. If it's a permanent block, then you can't activate it again. If it's a temporary block, then you can.
What about from the trauma they are getting like?
Well, there's different types of paralysis. This is just one type. You can have nerve damage, and that's pretty permanent. So there's different types of paralysis. If you want your muscles to relax, you just want everything to happen in reverse. You want calcium to be removed from troponin, and that reverses everything. You want calcium to go back into the SR. You want all that sodium to be pumped out. Again, the focus here is anatomy. I want you to know that the T tubules are important for spreading the signal. The SR is important for releasing calcium, and therefore it's going to take it back in. And troponin helps regulate the interaction between actin and myosin. But in case you're curious, that's how it happens. You get overlap with contraction, relaxation, the thin filaments move back, the Z lines move out. Yeah. Questions on how contraction works, and the names of the filaments, molecules, how they interact with each other. Because one neuron, and this is called a somatic motor neuron, by the way. A neuron that controls skeletal muscle is a somatic motor neuron. Because a somatic motor neuron can branch and control multiple fibers, it controls this fiber, over here it'll control a different one, over here it'll control a different one. Because one neuron controls many fibers, we call this together a unit, a motor unit. If you activate this purple fiber, if you activate this purple neuron, this one neuron is activated, all the ones here in purple also contract. This neuron is activated, all the ones in purple contract because they're all controlled by that one neuron. The motor unit is activated. So this is divergence. Sorry? Yeah. By activating one neuron, you can activate many fibers. Activation of one neuron will cause contraction of many fibers. In some places, like control of your eye, you want very precise movements. So precision means you want one neuron controlling fewer fibers. So you only have like 2 to 3 here. But if you want lots of strength, you want all those fibers to work together. The best way to have them work together is to have 2,000 fibers controlled by one neuron. So, like for lifting heavy things like in your legs, you will have lots of muscle fibers controlled by one neuron. Again, you can see that here. Sorry, let's go down my screen. Axon branches, axon terminals. One neuron controlling many fibers, that's called a motor unit. Okay. Here's the first receptor that we're going to mention that relates to something besides skin. We've been talking about Meissner's corpuscles, Pacinian corpuscles, hair root plexus. Those are all found in the integumentary system for sensing touch. There's this receptor called a muscle spindle. There's this receptor here called a muscle spindle. In cross-section, it looks like this. You can see muscle fibers here in cross-section. Here's muscle spindle in cross-section. What the muscle spindle does, it detects the amount of stretch that your muscle feels. We don't want our muscles to stretch too much. That'll be damaging. Just like if you stretch a rubber band too much, it'll snap. That's really bad for our muscles. And it probably hurts if you snap yourself. But yeah, muscle spindles detect stretch. We want to regulate, we want to know how stretched out our muscles are. If our spindles get too stretched, they'll tell our muscles that they need to, what's the opposite of stretching? Stretching means your muscles get longer. We need our muscles to contract. It's why if you stretch this tendon, this quadriceps tendon, that's why if you hit your knee just right, you kick your leg out because you're contracting these muscles that are getting stretched. It's a reflex to protect your muscles. And that reflex starts with these receptors, the muscle spindles. What these muscle spindles also do is that you want to maintain some level of contraction in your muscles all the time, otherwise you'll just be a limp blob. That's what muscle tone is. Muscle tone is the baseline contraction of your muscles at any given time.
The muscle spindle itself is the dark band that's around.
Yeah, the muscle spindle itself, well, I guess more accurately, muscle spindle itself is this dark band plus the stuff inside. It's the whole thing. Yeah. These look like muscle fibers because they are, but they're a special kind of muscle fiber. we're not going to get into that. If you want to talk about it later, I'm happy to. So it's this whole thing. And this is what it looks like in cross section. It looks a little different in longitudinal section. We have slides in longitudinal. So muscle spindles help with reflexes so you don't overstretch. Also just help you maintain tone so you're not a blob on the floor. So I get a lot of questions on what's actually happening when you're building muscle or losing muscle. We talked about how, let me go back to we're on slide 29. To form muscle in the first place, where was it? To form muscles in the first place, we have individual myoblasts that then grow and link together to form one really long muscle fiber. That's in early development. What about now as adults? What happens to our muscles if they get damaged or if we want to grow our muscles? If we damage our muscles, we just repair them. So there is repair to muscles. When we grow muscles, what happens is our muscles get a signal that they've been damaged. When we work out, we're slightly damaging our muscles. They get slightly damaged, the muscles grow in response. They'll grow in size. Each muscle fiber gets bigger and bigger in diameter. What about it is increasing in size? You're getting more mitochondria. You're getting more activity from those muscle spindles. So muscle fibers increase in size. You're not undergoing mitosis. These cells are not growing in number. They're growing in size. The term for that is hypertrophy. Hyper means elevated, more. Troph means grow. Hypertrophy.
So you're growing the muscles by adding more mitochondria?
Adding more mitochondria and other things inside of the cells. I'm just highlighting that because it's important. The key thing here is that the cells are growing, you're not growing the number of cells. Keep the same number of muscle fibers.
The cells themselves.
The cells, the fibers
Like
grow in
adipose.
size.
So,
Well, like adipose. Adipose, they grow in size when you gain more weight, you get more fat. So atrophy, that's without growth. Whenever you see an A or an N in front, atrophy is without growth. That's when the fibers get smaller. Here's normal cross-section of muscle fibers. Here's an atrophied muscle. Sorry? Yeah, it can be damaged or unused. Let's say you're bedridden, let's say you're injured, and you don't use your muscles for a long time, your muscles shrink. Yeah, even if they're unused. So again, it's not that you're not decreasing the number of cells, you're decreasing the size of the cells and the tummy.
Is that like what? Is it ALS?
With ALS, that's a complicated condition. ALS is a nervous system condition. If you don't stimulate your muscles, the muscles don't get fed. And so if you don't stimulate your muscles, they get weaker. So not only can you not control your muscles in ALS, you also get muscle atrophy. You get both.
But how long is it gonna take to produce like a normal?
How long does it take to get back to here?
Yeah.
I don't know exactly. I just know that if you don't use your muscles, like say you're bedridden, like you're literally not using your muscles. If you're bedridden for like 9+ months and you don't get moved around, you get permanent atrophy. You can reverse the atrophy if it's before that.
After permanent,
Then you're gonna have permanent damage to your muscles after 9 months of unuse.
like
If we're all mobile, if we're all able-bodied and we're moving around, you move your muscles a little bit, so you're using them a little bit. I'm talking about just no
like
use.
when you're in a wheelchair, like
If you're in a wheelchair,
you
yeah.
see people in a wheelchair with their paralyzed from the legs down, how thin their legs are, and it's because their muscles have been
Well, let's say you keep your hand in a cast forever and you never take it out. Then
what
you
about
have severe
like
atrophy.
during the pregnancy, women are losing their flexibility of the muscle. Are they gonna be again, like they are
tendency, what happens with muscles, I mean, in that region, you're going to get stretch of muscles.
They are getting thick and they are getting separate.
So there's some damage, so to speak, there. They're getting stretched out. They can be,
Is it just interesting? Is it permanent?
no, that's not, I mean, like, the changes will happen to those muscles, but you can get strong again. You can reinvigorate those muscles again. All right. When we look at muscles in cross-section, they don't always look the same. Some muscles in cross-section are smaller in diameter, so like here's smaller in diameter, here's bigger in diameter. So muscle fibers in cross-section can be different sizes, and they can be different colors, different tones. Here you can see that this is darker. You can see that this is lighter. This is smaller diameter. This is larger diameter. Here you see bigger gaps between the fascicles and even between the fibers. Here you don't see as many gaps. The difference between these two, the terms for these are fast twitch and slow twitch. The official terms are type 1 and type 2. If you call them fast and slow, I'm fine with that. It's more intuitive.
Which one is it?
Slow twitch is over here. Slow twitch is darker. It's darker because it has this protein called myoglobin. What does that sound like? We've heard that root globin. Hemoglobin. Hemoglobin is found in the blood cells, red blood cells. Myoglobin is found inside muscle, and it has the same function. Hemoglobin holds on to oxygen, so does myoglobin. This tells you that if you have myoglobin in these cells, they use lots of oxygen, because they have a reserve of oxygen on it.
It was in TED, the last discussion. It's very interesting. Can we increase the slow twitch fiber?
Can you increase the slow twitch fibers? What you can do is you can make them more efficient.
Or it's
I
like
don't know if you
genetically?
convert.
I think you're genetically like born with like
Certain
higher
proportions.
concentration. I remember this from a biology
Yeah,
class
yeah.
a couple
I
years
believe
ago,
you're
like-
born with certain proportions, but you can definitely work on your slow twitch. You can work on your fast twitch. You can make each one.
But it's like maybe, I think I read 60% is genetic.
I don't know the percentage, so I'm not going to say. I don't be wrong. But yeah, you can at least work on these muscles using different activities. You work on slow twitch, like with running, fast twitch, sprinting. These are strong, short bursts of energy. This is long-term endurance. Since they're for endurance, and since they're for endurance and not for strength, that's why they're smaller in diameter. Smaller diameter means they're not as strong compared to these ones. And they have extensive blood supply. That's why you see all these gaps. This is where capillaries would fit in. So this is how you identify slow twitch. Fast twitch. Oh, sorry, I didn't finish talking about it. You're fine. To help with that aerobic respiration, you also find lots of mitochondria. There's way more mitochondria inside. You don't see it on this, it's really small, but there's more mitochondria inside of these slow twitch. These muscles are important for anything needed for endurance. Muscles for posture, because you can sit like this for a long, long time. You can walk for a really long time. Not with the eyes or hands, and you can, if like an extreme example would be marathon runners, they have a really high amount. Fast twitch, type 2, this is for short bursts of energy, really strong short bursts of energy. And being larger diameter or stronger, downside is they don't have as much, they don't have as many capillaries, as many blood vessels leading to them, so they don't get as much oxygen, so they don't last a long time. Good for short bursts of energy. So like Usain Bolt can run however fast. He's got really well-developed fast twitch fibers. He can't run that fast for that long. He can do it with that for a short amount of time. Powerlifters can lift up a lot of weight for a short amount of time. They can't just hold it there forever. And I'm sure they can lift it longer than I could, but they still can't hold it for that long. It's short bursts of energy. Right, and so they're paler in color because they don't have as much myoglobin. Most of your muscles have a mixed content. So you'll have some that are slow twitch, some that are fast twitch. There's even an intermediate one, which we're not going to get into, which is, intermediate, as the name suggests. And so most of your muscles have a mix of them. Most of your muscles are not just one or the other. But good examples are your eye muscles or your leg muscles, but the ones used for, like, say, squatting or sprinting. Okay. So hopefully you remember this animation and what everything applies to. This is our neuron. What's this region called? Axon
terminal.
It's the axon terminal. Here we have neurotransmitter being released by exocytosis. What is this region called, the whole thing? Here's the neuromuscular junction. The receptors are found on the motor end plate. Here's the motor end plate. When the neurotransmitter binds to the motor end plate, we get a rush of sodium into the cell. That's what this flash is. When the rush of sodium comes in, it goes down the T tubules. Here are the T tubules on either side. And then here, this is this membranous thing that surrounds the sarcomere. What's this membranous thing in this pinkish purple? Sarcoplasmic reticulum, which releases these white dots. What do you think these white dots are? Calcium ion. Calcium ion binds to which protein? Troponin, which moves tropomyosin out of the way, which allows these myosin heads to grab and pull on the actin. And so you can see the Z lines moving in towards the M line. When you have a cramp, what's happening? I mentioned how it's like a sliding foot theory, like we have a really good idea what's happening, but we're not completely sure, we're not 100% sure. Same idea here, we're not 100% sure. Cramping likely happens for a number of reasons. The ion balance is messed up. Calcium, potassium, and sodium ion balances has to be just right for these to work. Could be lack of glucose, lack of ATP, something about phosphate being attached to the myosin heads, which why is eating and cramping a workout a thing? This is off topic, but the short answer is, like when you eat, you often cramp. If you're eating, you're activating your parasympathetic nervous system, so you're sending, one thing that does is sends blood to your digestive tract. You want to absorb nutrients, which means you're taking blood away from your muscles. You need more blood to your muscles if you want to work properly. If you have lack of oxygen, lack of glucose.
Binds with the myosin?
Calcium binds to troponin. Calcium binds to troponin. If you go to Crash Course, it has a really good animation of all this.
Yes.
Sorry? That's why you pause it. It's a video. You pause it. Pause, rewind, pause, rewind. Just like when you read, I can't just read straight through. I have to like go back and reread. Same thing. Sorry, do you have another question, Jess?
Okay, I was just saying, so after the calcium binds with the troponin, then that sends a signal to the tropomyosin, the other two ones, tropomyosin, to move out of the way, and then that's what allows the heads.
When calcium binds to troponin, you can see how troponin is also attached to tropomyosin.
Yeah.
So when calcium binds to troponin, troponin moves and pulls tropomyosin out of the way.
Okay. And then when that is out of the way, the heads of the myosin can latch onto the actin.
Yeah, then myosin can bind actin. Exactly. Take a moment. Raise your hand if you need more time. Answers on 3. 1, 2, 3. 3 is the letter. 1, 2, 3. D. D. Yeah, motor end plate, that's where you find the receptors. Axon terminal is across it. Let me actually point something out to you real quick, because it's super confusing for people. This one. We have two models of skeletal muscle. This is a muscle fiber, and so on this one, here's the neuron in yellow, and we'll get to what this is later from Island. Here's the neuron in yellow, so this is the axon terminal. This wrinkly thing, that's the motor end plate. So it looks weird on this one. Wrinkly thing, motor end plate, yellow thing, axon terminal. This one, also showing the muscle fiber. Here's the axon. These blue things, this blue thing is the axon terminal. You can see it's bulbous here. It's bulbous here. It's been cut off right here. The reason why it's been cut off is so you can see the motor end plate underneath it. So this is the motor end plate. The blue bulbous thing is the axon. Together they make the neuromuscular. All passes around. Where the signal is released, this is detecting the signal. Muscle spindles detect stretch. Okay. Does everyone have their packets? Raise your hand if you don't have the package. Okay. Here's what we're going to do. I have clipboards if you need one, so grab a clipboard, and we're going to walk out. We're going to walk outside just like last time to start our lab check 7. The 4 most important things that you should have down. The 4 most important things that you should know like really quickly are your muscle's name, of course. Okay, maybe 5 things. Muscle's name, its shape. When I say shape, remember it's one of 3 things. It's either parallel, pennant, or circular. You fit into one of those 3 categories. Pennant, parallel, or circular. Sorry? Movement, you might have more than one movement. You cause at least one movement, that's the job of a muscle. But we went over terms like elevation, depression, protraction, retraction, abduction, adduction. It's going to be at least one of those terms, possibly more than one. You should know what your muscle does there. You should know what body region your muscle is located in. Remember, use the anatomical term. And origin and insertion, I'm looking for the bone feature. Like, are you attached to the curving process, to the coracoid process? What are you attached to? What are your attachments? Those you should have down, and you should be able to say quickly to the person you're meeting. Do you need a minute to get that down? I'll give you like 3 to 5 minutes. I'll turn the lights on. Hey. If you have any questions, if you want to double check anything, come ask me. Yes. Which one is it? You actually put their hand up. Come around, 1 second, 1 second. Come around, come around. It's gonna be.
So I have 3 months in
Where are they?
the transfers they have so many
Is there a general region?